how are you?
Hi, nice to meet you. I honestly am just feeling awful between all my homework for my bio and chem classes and soccer practice literally every single day. I just gotten no sleep and now I feel cold all the time and my neck is super tight and it's just been terrible.
Oh, Sue, I'm so sorry you're feeling like that. We're doing our best to figure out what pathogen is making you so sick.
How are you going to do that?
We use a process called polymerase chain reaction, which is also known as PCR, which is used to amplify the DNA of a pathogen.
Oh, I think I learned about that in bio class. Isn't that when the DNA is heated so that it can be denatured, and then it's cooled during annealing so primers can attach, and then during elongation, free fluorescently tagged nucleotides are added to the DNA?
Yes, wow, that's exactly right. PCR must be used in the cycle sequencing process so that a large number of copies of DNA that differ length by a single base can be produced. And so they all end with a fluorescently tagged nucleotide, which is caused by dideoxynucleotides, creating these fluorescently tagged nucleotides. And so that causes the reaction to stop. And so then the result is a bunch of different strands of DNA that all differ in length by a single base. And so these can be placed into an automated sequencer. And so, however, PCR can also be used in regular DNA sequencing, which uses the same methods except that in the elongation process, identical copies of the original DNA strand are produced.
Oh, I see now. So then the strands are digested by restriction enzymes to create restriction fragment linked polymorphisms, or RFLPs. And then these can be analyzed during gel electrophoresis because when they're run through the gel, the smaller fragments run faster because they have less resistance. And this is used for things like genotyping because it can be used to identify a specific gene, or it can also be used in things like paternity tests or identify suspects at a crime scene.
So yeah, that's exactly correct for regular DNA sequencing. But with cycle sequencing, we place our different DNA strands into thin capillary tubes with buffer solution that go into an automated sequencer that performs gel electrophoresis on them. And so a laser beam picks up on the fluorescently tagged nucleotides and sends this information to a computer where you have a graph with peaks that are different colors. And so scientists can identify these peaks and then put this information into BLAST, which is a database for identifying pathogens, and match them. So that's what we're doing with you.
Oh, so that makes sense for bacteria because they always have DNA, but what about other pathogens like viruses?
So with viruses, it's a bit trickier because they often have several more errors in their DNA and RNA sequences. So, however, because all pathogens contain DNA, we can sequence all of them. So with viruses, this means that before you begin PCR and the DNA is isolated, scientists must use their hypothetical diagnosis to know the anatomy of the viral DNA sequence, and this ensures that they cut the DNA at the correct sequence so that they can get a good diagnostic information. After that, the DNA sequencing process is identical.
I learned in bio class that viruses have small genomes, so it makes sense that PCR is used because they can amplify the amount of genetic information that you have to work with.
Yes, exactly.
Here are the tests that you ordered, Dr. Ava. They're for the patient Sue Smith.
Thank you so much. Wow, okay, so now we can finally diagnose you. So you have Neisseria meningitis, which is caused by the meningococcal bacteria. It's a relatively rare disease, but it is severe. So for the Neisseria meningitis infection, I'm going to be prescribing you some antibiotics, probably going to be penicillin if you're not allergic, or ceftriaxone. And you might have to stay in the hospital for a couple days because it's slightly serious. I know, I'm really sorry, but you're not going to be able to go to soccer for at least a week until your infection decreases, because this is, if this progresses, it can cause death.
So how did you figure out my diagnosis?
Well, like I was telling you before, we used our cycle sequencing process, but we also knew before that, based on your symptoms of a 100.6 degree fever, headache, extreme lethargy, and a stiff neck, we hypothesized that you had a form of meningitis, since those are the same symptoms. So we took your blood, fluid, and lymph samples to the laboratory, right, where they tested it, and they used cyclosequencing. And so when we analyzed, this is Sue's results.
Oh, look
Yeah.
at that.
Yeah.
It's so pretty.
Yes. So what we did is we analyzed this, and each of these colored peaks has a different nucleotide that we were able to identify, and we put this in the blast, and it said that you had Neisseria meningitis with a query cover of 100% and a 96% identity when compared to that strain of meningitis. Sue, I'm so happy that you came to us because we're going to treat you and we're going to prescribe your antibiotics. You'll be out of here hopefully in a week if nothing bad happens. And so it was nice to meet you.
Nice to meet you too. Thank you.
What are you doing, guys? Okay, we'll get them around.
Lord, oh Lord. Hi Sue, I'm Dr. Ava. How are you feeling today? Wow, okay, so now we can finally diagnose you. So,
Now we can finally diagnose you.
yeah.
Oh. It's Dr. Ava. Get that one out.
Okay.
And you feel like falling down, all care